From Microbe TV, this is Office Hours. I'm Vincent Racaniello, and today is March 8th, 2023. You know, every time I say from Microbe TV, I want to make an episode number, right? Because I'm so used to doing that with the other shows. But there's no episode here. There's just me and you, folks. Welcome, everyone, to our Wednesday evening office hours. And I would like to thank our moderators for coming tonight. I saw Les, I saw Peak PDK, I saw Barb Mack, I saw Frank, and I said Les already, right? And I think Tom is going to be coming in late. Welcome, everyone. So this is Office Hours, which is named after, of course, what professors do in courses once a week or so. They have a set aside time where students can come and ask questions. In the old days, it was actually in my office. I should show you a photo of it. I have a really good photo. Let's see if I can bring it up. What lecture would that be in? What lecture would a picture? Oh, the plaque assay lecture. Yes, of course. So let's go to that. Since this is office hours, we can bring up our lectures. So the plaque assay would be in the infectious cycle. I believe that's lecture number 3. No, that's not it. Lecture number 3 is genomes and genetics. Must be lecture number 2. It's a cool picture, so I want to show you. The real office hours, which now is on Zoom, because I don't have an office anymore. Here we go. I very much like this photo taken, I don't know, a couple of years ago. So let me turn on the sharing here. Sharing. I do need a producer to do all this. Okie dokie, here we go. So that is, let's make it a little bigger. Scroll it down. Yeah, that's my office a couple of years ago. And these are 1, 2, 3, 4, 5 students who came. This was the final office hours of the year. It was before the final. And that is the wall of polio behind them. And it was a nice, good bunch of kids. I had a good time with them. And these are the ones that were devoted. They came often to the office hours. They came to the last one. And, of course, behind them is the wall of polio, which I just left behind. As you may know, I shut my lab last year at Columbia, and a few weeks ago I emptied my office because I was asked to vacate it, and which I did. I threw out most everything. I left the wall in place. I let the next person take care of it because it's crazy glued to the wall, and I didn't feel like dealing with it. So anyway, that's office hours, and you're welcome here, and you are welcome to ask whatever question you'd like about viruses. I'll do my best to answer it. Some of them I'll say, I don't know. I'll find out. Don't give medical advice. If you want medical advice, I can pass it on to Daniel. Actually, I am doing TWIV clinical update right after this tonight. So I can always pass on your questions to him. Couple of items before we get going here. So next week, Vanity is just calm. Thank you, Vanity, for moderating tonight. Next week is actually spring break at Columbia. And I will be in Atlanta doing a TWIV at a laboratorian, a clinical laboratory type setting, and it's a meeting for clinical laboratorians. Okay, and I don't want to do office hours for my hotel, and so I have decided to take the prerogative of spring break and cancel office hours. So next week, whatever that is, March. Let's look it up. March 15th, no office hours. We'll resume the following week, with Q&A with A and V. That's March 22nd will be Q&A with A and V, okay? Couple of other things. If you'd like to donate, you can use the Super Chat button here. That's fine. You can also use Venmo, which takes less than YouTube does. Okay? YouTube takes a big chunk. And I love seeing your donations here. It's wonderful. But if you want to have more, go to Microbe TV. Here we go. And that's the Venmo address. And also, we are doing, of course, TWIV episode number 1000 on April 15th in New York City at the Helen Mills Theater. Very exciting. 6 p.m. It's a Saturday night. Live from New York. It's Saturday Night TWIV. How about that? The entire TWIV cast will be there. The Randy Despommier Quartet will be playing before and after. And we will all have things to say to you. So there are some tickets available yet. They're not limitless. Okay, we have sold over almost 110 tickets, so there are 20 left. I think 110 is great. And we're asking, of course, for a donation of $100. We're asking for a donation of $100 minimum to defray our expenses. It's rather expensive to rent a theater in New York City, but it's a few blocks from the incubator, so afterwards we can all go there, and we're planning to have a little reception. And the Randy Despommier Quartet will play there as well. So if you are interested, send me an email, vincent@microbe.tv, to reserve your ticket. You will get a ticket PDF in the email, and you bring it with you, and you will show it at the door. Someone will be there. Okay, so that's TWIV 1000. And in fact, I made notes today in my field notes, right? And I'm reading them today. So I got my stuff together tonight, at least. I don't know if it'll be together every week. Oh, here's another cool thing. Where is the thing? Here it is. This was a gift sent to me by Lise, who's here tonight. Thank you, Lise. It's a Professor Racaniello bobblehead. Let's get that in there. Yeah, which I got yesterday. I think it's pretty good. The glasses aren't right. It just makes me younger looking. It's cool, right? That's never bad. So thank you, Lise, for sending that. And a lot of people have said they would like to buy one. Okay, I don't know if you're true or not. I'm going to buy bulk. I'm going to buy 50 in bulk, so you can buy. So I don't know how much they're going to cost, but hopefully in bulk they're less. And so if you want a Racaniello bobblehead doll, you can buy one. If you don't, that's fine. And I'll just have a whole box full of them sitting at the incubator that nobody wants. That's fine. Yes, thank you, Lise. Thank you so much for that. Very kind. I was very surprised to see that. It did bring a smile to my face. It's cool. It actually kind of looks like me, right? I mean, it's not perfect, but what the heck? I like that little spiky hair. It's cool. It's a good haircut. No gray hair, no gray beard. Good. It's good stuff. All right, that's the other thing. Okay, bobblehead photo, TWIV 1000, no office hours next week. What I wanted to do is remind everybody that we have, let's get rid of the students there. The Amy papers, I've been updating these, right? And I put a bunch of papers in today. Yeah, you think the bobbleheads will sell out, huh? Okay, we'll see. I don't know. I don't know why you'd want a Racaniello bobblehead, but hey, I'm not arguing. We'll sell them. If they come in time, I could sell them at the TWIV 1000. Otherwise, we'll mail them out. Anyway, these are some new papers here, and I wanted to pick out two of them for some brief discussion. The first one is this one, which was published in Nature Communications. Maternal SARS-CoV-2 vaccination and infant protection against SARS-CoV-2 during the first 6 months of life. It's pretty cool. And this Daniel. I'll get back to the bobbleheads. Let me get through this first. Daniel talked about this last week. So this was infants born at Kaiser Permanente, Northern California, and they looked at 2 or greater doses of vaccine received during pregnancy during months 0 to 2, 0 to 4, and 0 to 6 of a child's life. This is during the Delta variant period. Remember, this is an ancestral vaccine, so it's against the original isolate of SARS-CoV-2. So greater than 2 doses, 84% effective during months 0 to 2, 62% during months 0 to 4, and 56% during months 0 to 6. This is any kind of COVID, right? It's not just severe disease. And then they looked at hospitalization of infants. Over the entire study, the incidence of hospitalization was lower during the first 6 months of infants of vaccinated mothers compared with infants of unvaccinated. That's 21 per 100,000 person years versus 100. It's about a 5-fold reduction. But this is vaccinating the mom, right? So you provide 6 months of protection, and then at 6 months, of course, the child can be vaccinated. So I think that's pretty cool. I like that. We're starting to get some, in my opinion, some interesting data. All right, so that's that. You could check that out. It's open access. And then we have this one. So I've always been critical of long COVID studies because they're observational and these symptoms are vague and self-reporting. But this one, again, Nature Communications, long-term gastrointestinal outcomes of COVID-19. So we focus on one set of symptoms, which I like, right? Focus on one set of symptoms and gastrointestinal symptoms. And this is, again, a Department of Veteran Affairs, excuse me, my alma mater is calling me for a donation. Come on, Cornell. Don't you know I'm live streaming? Department of Veterans Affairs net database, okay? So we have 154,000 people with COVID and 5 million controls and 5 million historical controls. And they show beyond the first 30 days of infection, people with COVID have increased risks of incident gastrointestinal disorders like motility disorders, acid-related disorders, functional intestinal disorders, acute pancreatitis, hepatic, and biliary disease. And they are evident in people who were not hospitalized, okay, and increased in a graded fashion across the severity spectrum. So non-hospitalized, hospitalized, and admitted to ICU, you get more intense gastrointestinal symptoms. So I'm convinced that there's something going on here. There are some potential issues with this. First of all, it's a VA study, right? A certain population. It's mostly men. So there could be differences in the general population. But I think focusing on one set of symptoms is useful. Now, what they show is hazard ratios here and excess burden per 1,000 people. And these are all the different diagnoses here. And you can see the hazard ratio, it's between 1 and 2, which is 1 to 2-fold higher risk of getting these disorders after COVID. And the excess burden per 1,000 people, 1, 3, 10, 20 per 1,000. So those are the number of people who are getting GI issues after COVID. So they don't know what the effect of vaccination is. So I think we have some work to do here. But this, I think, tells us that at least for GI symptoms, we have issues. And it's going to be a large number of people that have this, and we don't know how long it's going to continue. So they're going to require quite a bit of health care, right? All right, so people are saying, why don't you make bobbleheads for everybody? Yeah, that's a great idea. Somebody wants to do it. I don't want to go back and forth with photos. It took Lisa quite a bit of work to get this done. They have the thing for me. Um, you know, to make 8 different bobbleheads, I'm not going to make 50 copies of 8 different bobbleheads. I mean, this company doesn't do fulfillment, right? So I have to buy them in bulk and sell them. I just don't want to do that. It's just an excuse for restricting it exclusively to me, at least for now. We'll see how they do. Okay, if they sell well, we'll consider it. We'll do one at a time. How's that? Starting after me, it'll be Dixon. And then it would be Alan, and then Rich, and Kathy, and Brianne, and then Daniel Griffin would be cool, right? And then Angela. So Angela's barely been. So I don't know how many people know her. So anyway. The other thing I wanted to discuss is the Department of Energy report, right? Which we don't have. What we have is them saying they're convinced it came from a lab with low confidence. So I've been corresponding with Eddie Holmes and Bob Gary, and they say, yeah, we don't know what they have. We may never know. The panel today that they had a House hearing, this is completely stacked against reason and logic. I know 2 of the 3 people. Someone type in the third one. I know it's Robert Redfield, former CDC, who botched COVID during the pandemic, right? Nicholas Wade, former Times science writer who was moved to the dark side. I don't know who the third person was. I mean, where are Eddie Holmes and Bob Gary and Christian Anderson to give the other point? You just stack it because you want one outcome. So I'm not convinced they have anything. So you should ask me, Vincent, what would they need to have to convince you that it came from a lab? I don't even know what lab they're talking about now because, you know, the Wuhan Institute of Virology is 8 kilometers across the river from the Huanan seafood market. And the Huanan seafood market is clearly the epicenter of the early outbreak, as shown by Michael Warby and colleagues. So there's this Wuhan CDC next to the Huanan marketplace, but there are no cases there. They're at the market. What would I need? Well, so here's the thing. I think they're saying it likely came from a lab because we haven't found the ancestor in nature. And that's just disingenuous because we have highly related viruses in nature. So clearly similar viruses, very similar to SARS-CoV-2, especially in the receptor binding domain, hugely similar, are circulating out there. So it shows that it could have come from nature. We have phylogenetic trees showing those relationships. We have the early epicenter at the market. And we have, of course, the historical fact that every human virus has come from an animal at one time or another. Okay, you want to ignore that and just say this could be the first? Okay, that's fine. We'll ignore it. We'll ignore history. You know, that's a good practice in general. We have data indicating a likelihood of a zoonotic spillover, but no data saying it came from a lab, except that the CDC was nearby. Really? That's your data? So what else do you have? If they don't tell us, we can't fight back. And these hearings are baloney because they're stacked in favor of people who think it came from a lab. So this is just a ploy. It's horrible. And I don't know that they will ever release it, but they do have, because they don't have anything. They don't have anything. Has David Quammen voiced an opinion? I haven't heard. I haven't. If you ask David, he will waffle because he wants to look into it first. He wants to see what they have and so forth. So I could email him. Sure, I could do that. But Eddie and Bob Gary have been emailing back and forth with me, and they just say it's just a whitewash. Is that not a whitewash? It's just throwing junk at the screen, and they can convince people that hello Tom, welcome and thanks for moderating. They can convince people because the Department of Energy, the FBI, they say, oh, it's a government agency, they must be right. Really? Seriously? Well, tell us what the evidence is then. And the other thing is that this is not something you solve with an investigation. You have to get data, and I don't know how they would get data. They're not going to get into China and get data. They're not going to have isolates. They're not going to have phylogenetic trees. It's just an agenda, and it fits their narrative, and that's all. So when we get the data, we'll look at it. But until then, I can't say anything. And all I can say is we have data supporting a zoonotic origin. That's quite good. And if you choose to ignore it, then you're not following science. So put your thoughts in if you'd like, and I'll get to them. How can one hope to find ancestral virus 3 years later? Elena writes. Well, ancestral SARS-CoV-1 was found 10 years later. Well, the parts for making SARS-CoV-1 were found in bats in a cave in Yunnan province. No, where was that? I forgot where the cave was. So you can do it, but not much longer, because yes, the viruses continue to reproduce at high rates and they change, and you're not going to recognize them anymore. And let's see, can the lab leak and spillover both be true? As in they were studying something from the wild in the lab. Yeah, but they weren't. Wuhan Institute of Virology, as Eddie Holmes has said, and yesterday he put up a Twitter thread with all his data. They published all the viruses they had, and SARS-CoV-2 wasn't in it. This was before the pandemic. So why wouldn't they publish SARS-CoV-2 if they had it? Because they had no idea it was going to cause a pandemic. They would have published it. And also you have to understand that there were at least two spillovers at the market, because there are two lineages of SARS-CoV-2 with slightly different genome sequences. And so, a guy from Fox News says, well, couldn't somebody from the Institute have gone shopping at the market? Well, yeah, but that's not... If they were infected and they brought it to the market, it's one lineage. Where's the other coming from? And why weren't there cases then at the Wuhan Institute of Virology? There weren't, by the way. Don't believe what the Post wrote. There are no cases of COVID at the Wuhan Institute of Virology. That's nonsense. So, all right, when I get to more, I'll address it. But let's hit some. Hello, everybody. Nice to have you here. I'm not going to go through where you are, because I know where you are. You're mostly the same people. Welcome back. And I don't know what this means. John writes, any idea what's up with Robert Redfield? Is there any data to support this? So what he said today? I don't know what he said, but I have the statement from Wade, because it was published and I have it. But there isn't really anything there. I know if there are data, I'm happy to look at any data. You know that. We're scientists. But I don't see anything. They haven't put anything out there. Kang, hello. How are the various flu viruses identified? Are these serotypes in each type considered a variant? If they are epitopes, why can't they be used to immunize? Okay. Well, nowadays, if you have someone with suspected influenza or an animal, you're just going to be screening. If they have a weird illness, like if it's birds and they have a flu-like illness, then you would do genome sequencing, and you would identify the virus simply by genome sequencing. And the H and the N refer to the hemagglutinin and the neuraminidase proteins. And there's so many hemagglutinins and so many neuraminases. And H1, 2, and 3 hemagglutinins and N1 and 2 neuraminidase infect people and cause epidemics and pandemics as far as we know. And that's it. The others are in animals. So H5, infects people sporadically, but it doesn't transmit. There have been like 800. I have the WHO numbers here of H5N1. Here we go. From 2003 to 2022, 865 human cases, 456 deaths. And the big ones are Egypt with 359 cases and Indonesia with 200 and Vietnam with 127. And these are countries with a lot of poultry and other birds, right? So right now there's an H5 vintage, let's say. So these viruses undergo antigenic variation. They can change from year to year. And if you want to make a vaccine, you'd want to match it to whatever is circulating. So the U.S. does have an H5N1 vaccine stockpiled, but it's not matching the current circulating one, so you'd have to make a new one. And people are doing that, of course. They're making H5N1 vaccines, making mRNA vaccines, which you can do relatively rapidly. So you would use, in the case of, if you want to make an mRNA vaccine, you could put the H5HA in it. That's it. So that's ongoing. Now, this is funny. My kid just had a virus, threw up all over the car. They tested and it was negative. Well, if you throw up once, there are a lot of reasons you could do that. Viruses will make you throw up multiple times. And so I'm thinking of norovirus. But, just once, there are a lot of reasons to throw up. So it doesn't have to be a virus. Anybody watch the hearing? So I didn't watch it, but if anyone did, give us a summary. And your summary can be, it was nothing. How about a live stream with Lori Garrett? Yeah, I think she might be interested, but I don't know if she wants to do it from 8 to 10 p.m. I could ask. I have her email, no problem. Okay. Going through the early comments here where everyone's just kind of bantering. Michael Warby gave a lecture on the myth of the lab leak hypothesis. Okay, that's good. Everybody needs to speak out who can to cover all the bases. Yep. Nicola, hello. I finally got around to watching the virus lectures. Can you define what 3' and 5' are and why they are called that way? Yeah, sure I could. Let's see if I can get a nice picture to illustrate this. Just give me a second here. Yeah, maybe this is a good one here. This is a picture of a base, okay? All right, let's turn on the share. We'll turn off Nicola. Okay, so that is a base. And the carbons are numbered. And you see 1', 2', 3, 4, 5. And so the 5' end would be up here, the 5' end of the DNA, and the 3' end is down here. So if this were the first base, that would be the 5' end, and if this were the last base, it would be the 3' end down here, because the bases, which comprise a sugar, this is a ribose sugar, and it's attached to a base. There are 4 different bases, right? And then there's a phosphate in between each of the bases. The very one at the very 5' end is a 5', and the one at the 3' is the 3'. So it's a chemical definition that makes it 5' and 3'. So, for example, DNA is synthesized in a 5 to 3' direction, but it is the template is copied in a 3 to 5' direction. So the polymerase is moving from 3 to 5', and it's synthesizing in 5 to 3'. And you know, many students get that wrong, even though it's rather straightforward, right? Many students get it wrong. It's a little bit confusing. Okay, Peak has a question about during SV40 replication. Does one hexamer fall off early and allow the other one to finish splitting the genome, or do the two hexamers collide? Oh, I need a picture for this. Yeah, let's go to the pictures. This is true office hours, folks. That's DNA replication is lecture. What is it? I think it's 7. Let's see. This is a good opportunity for everyone. Nope, that's transcription. So it would have to be lecture 8. It's a good opportunity for everyone to brush up. You know, we're up to lecture 14 in the course now. We're at spring break, right? We just finished adaptive immunity today. All right, here we go. We're going to move into pathogenesis and vaccines and evolution. It's very exciting. So I know you have better things to do, but listen to them again if you've seen it already. It really reinforces it. So the way SV40 replication works, SV40 is a double-stranded circular DNA right here. It has a single origin of replication, which means that's where DNA synthesis begins. And the way it begins is that large T, 2 hexamers, 6 copies times 2 of large T, bind at the origin. There's the origin right there. They bind at the origin, and they denature the 2 strands. They separate the 2 strands, and then DNA synthesis occurs. And this bubble gets bigger and bigger, right? So here it's getting bigger and bigger and bigger. And Peak wants to know what happens at the end when they collide. So here is one of the hexamers moving along the replication machine. There's going to be one on the other side as well. And I don't think we know the answer to that, right? Do they collide and fall off? Does one fall off and let the other go through? Don't think we know. It's a good question. And, you know, there's still some people who are studying SV40 DNA replication. I could actually ask them. But I suspect, so this is one of those details that I don't think has been worked out. Okie dokie. Sad to see you being attacked on Twitter by lab leakers. Yeah, of course. They're all just dorks. They're such dorks. It's unbelievable. So I put up attack. First of all, people can't just have a nice argument. They have to attack you, which is why I go to Twitter. I drop things to antagonize people, particularly anti-vaxxers and lab leakers, and then I leave. Right? Hit and run. And so then I dropped 3 things the other day. One is Eddie Holmes had a nice thread about why the lab published everything they had, and it didn't include SARS-CoV-2. And then I said, if you want to listen to it, here's the TWIV episode. And then I said, you know, the Post just published this article, the Washington Post, saying what really happened in those early mysterious COVID cases in Wuhan. Is China holding something back? Actually, they're not holding anything back. And there's a wonderful epidemiologist in France who made an analysis of it, and their conclusion is based on an error. So I put the link to that up as well. And of course, I knew people wouldn't like it. Do you think I'm going to sit there and read through the comments and rebut them one by one? I have better things to do. I got them all fired up so they waste their own time. But thank you for telling me, Simon. Yeah, people love to attack. Oh, absolutely. I get attacked by email, too. Oh, all kinds of stuff. And then in the YouTube of TWIV that went down Friday, someone said, "Oh, Vincent, data is a dimensionless word. You're not right to say it's plural." Oh, go away! If not dimensionless, what do you think we have datum and data for? He said, it's one thing to be snarky, but if you're wrong, it's bad to be snarky. I said, well, can't you just voice your opinion and don't call me snarky? Anyway, let's go on here. Folks, here's a good question. What causes Paxlovid rebound? There's no such thing as Paxlovid rebound. There's COVID rebound. I'll get a figure out. Since this is office hours, we're gonna get figures. So where would I have a nice figure? I think I showed it to the doctors the other day. I gave a virology lecture to MDs at Columbia, infectious disease pediatric ID fellows. Right? These are docs training to be docs. So let's see if I can find it. It would be CUMC fellows. Yeah, because I have in that lecture hundreds of keynote files with all kinds of lectures. And it's always a matter of finding the right one. So did I have this? Here we go. I'm going to put it up here. We're going to turn on the screen share. So there you go. So this is Daniel Griffin's Stages of COVID. You can find the original article down here. Note, 2 phases, the viral replication phase and the inflammatory period. Viral replication, you get exposure. TE is the time of exposure. The virus begins to reproduce, replicate its genome. The RNA levels rise. This is viral RNA as measured by PCR. It peaks, there's an incubation period of 2 to 14 days, and then you have symptom onset here at the peak of replication, but you're already shedding and transmitting before symptom onset. And then that first week, the viral loads go down, you recover, and if you're lucky, that's the end of your illness. But in many people, you have a second phase, an inflammatory phase, where you have an overexuberant immune response, and that can wreak havoc. And here is where you can't breathe, and you go to the ICU and so forth. That is COVID rebound, right? There's one phase, and then there's a second phase. Apparently you're getting better, and then you have this second inflammatory phase. That's COVID rebound. That's what Daniel calls COVID rebound. It's not Paxlovid rebound. It's not caused by Paxlovid. If you take Paxlovid, you may put down, lower the viral loads, and then when you're over the 5-day course, you may then have an inflammatory period. That's all that is going on there. So there's no Paxlovid rebound. And doctors who refuse to prescribe Paxlovid because they say, I don't want you to have Paxlovid rebound, they're not right. Paxlovid isn't causing any rebound. It's a natural history of the disease. So thank you for asking. Give me a moment to clarify that, right? Yes, so Michael Warby. So we have stuff early, we have it deep, and still we cannot get penetration in this world. We have it way before everybody else does. We have Eddie Holmes, we have Michael Warby, we have everybody here for an hour, which nobody else is going to give them, and I still have to struggle because we're too complicated, we're too long. Well, that's the point, folks. It's long, it's deep. You want all the information in one place. That's the way to go. Okay, if the polymerase interacts with PLP at the 5' template, and how does it read the template from? No, it doesn't interact with the 5' end. It starts at the 3' end always. It reads from the 3' to the 5' end, and synthesizes in the 5 to 3' direction. Do you want a picture? I think we have one here. It's not going to be in the doctor's thing. Let's see, where is there a good picture of templating? I think that could be in lecture 6. Yeah, because that's RNA synthesis. We have a good picture there. Hold on one second while I open the file and find the slide. I mean, I guess this is office hours, right? This is what it's meant to be. Yeah. Okay, synthesis of RNA from RNA. Let's get the picture. There we go. I think this will do it, although maybe you'll be a little confused. Hold on. Screen share. So let's look at the top here. The template. The black line is the template. There's the 3' end, right? Some polymerases need a primer. So here we have a primer in red sitting down, base pairing with the template. And then the polymerase is going to start polymerizing at the 3' end and adding bases in a 5 to 3' direction. And that's how it works. So you start at the 3' end, and at the primer you're starting at the 3' end as well. Okay, video upload 99. What exactly is the wall of polio? I showed you a picture of it. Let me show it to you again. Recent, what lecture was that? That was lecture 2, right? Yeah. Okay, here's the wall of polio. So these are 6 well plates. Do you know what a 6 well plate is? Probably not. So let me find a 6 well plate. I was accused of being impossible to understand the other day. So now I'm going to explain everything. Here we go. Screen share. I just turned off screen share. No, I don't want a subscription. That's a 6 well plate right there. So 1, 2, 3, 4, 5, 6. It's a single piece of plastic. It has a lid on it, and you grow cells on these dish areas. You put medium on top. So you can do 6 plaque assays here, right? So that's what the wall of polio is. All right, let's go back to the screen share and get rid of this 6. There it is. So that's the wall of polio. A stack of 1,600 6-well plates. It was one experiment done by a postdoc, which was to do a plaque assay of a huge experiment. So we needed a lot of wells, and so that's what that is. I stacked them up after they were stained and dried. I stacked them up and glued them to the wall and to each other, and made a nice wall. So why do I call it the wall of polio? Well, it is a wall, right? And the plaque assays were done with polio. However, there's an even deeper reason. I'm going to get into the weeds here. I tell you. Here we go. And some of you may know this. Look at this. So this is the wall of polio. Do I have this damn thing on? Yes. It's called the Polio Wall of Fame, a set of 15 sculptured busts of 17 individuals who made important contributions to understanding and preventing polio. They're down in Warm Springs, Georgia. So the Polio Wall of Fame. So I named mine after that. In my lab, we have a slightly different wall. We call it the Wall of Polio, a collection of 6 well culture plates that have been used to measure the concentration of poliovirus in various samples. I love it because it's a teaching moment, right? So that's the wall of polio. All right, thank you very much. If I remember correctly, in the early days of COVID, the popular view was that the virus wasn't mutating. Well, no, that's not correct. The popular view was that it was mutating quite slowly. It was mutating, for sure. All viruses mutate when they reproduce. There's no question about it. Some slower, some quicker. It was mutating, but very slowly. I remember that quite distinctly. And then once it started getting into millions and millions and millions of people, then we got more and more mutation because it just was reproducing more. And that's where the variants were popping up. So it's just a matter of infecting more hosts. And if you have a certain rate of mutation, if you say in one host, the virus sustains 10 mutations, So then if you've got millions of hosts, you have many millions of mutations. And so it's more likely that the ones are going to pop up that confer some fitness advantage to the variants of concern. Alcohol or soap for inactivating SARS-CoV-2 or influenza virus. Either will work with an envelope of the virus. You can use alcohol-based sanitizers and soap and water. Now, we always say that norovirus is not inactivated with alcohol-based sanitizers because it doesn't have an envelope. But someone sent us an email, which is going to be tonight on the TWIV clinical update. But I can actually show you the link here because you're here on office hours. So why shouldn't you get the latest information? Here we go. So this individual who's at Rutgers wrote, while it's true that conventionally formulated alcohol-based hand sanitizers have limited effectiveness against non-envelope viruses, including norovirus, there's research in the published literature which indicates that these products can be reformulated to have significantly greater efficacy. So here's the letter, right? So this is the notes where I read the emails, and at the end it goes, that's TWIV, Weekly Clinical Update with Dr. Daniel Griffin. Thank you, Daniel. Okay, here are the papers. Let's have a look at them. Improved inactivation of non-enveloped enteric viruses and their surrogates by a novel alcohol-based hand sanitizer. There you go. So you can buy this stuff. Okay, hopefully they tell you what it is. Let's take a look. Purell VF477, 70% ethanol with a synergistic blend of polyquaternium 37 and citric acid, as well as gelling and skin conditioning ingredients. So there you go. That's what they used, and they said it's better than conventional ones. But that's for norovirus. For flu or COVID, you can use soap or conventional Purell. Okay, let's see. Interesting, you did the mRNA persistence snippet on TWIV. I read the paper best I could after Dr. Soup. Yeah, I get it, Dr. Soup. We don't have to mention his name. Messed with it. Did you spot he implied humans might have the polymerase enzyme to copy mRNA? I didn't, but that's completely wrong. We do not have an RNA-dependent RNA polymerase. It's ridiculous. You can say whatever you want, right? Apparently. And you get millions of YouTube subscribers. You know, folks, I will never say something that is purposefully wrong just to get more subscribers. It is completely contrary to my training as a scientist. And so, you know, Dr. Soup is not a scientist, but there are many scientists who say wrong things. All these pro-lab leak scientists, they should know better. I don't get it. Anyway, yeah. We don't have an enzyme that copies RNA and makes RNA. No, we do not. I don't know the answer to this one. When a doctor does a spinal tap on someone with Lyme, what are they looking for? Don't know. No idea. When the CNS is suspected, then you do a spinal tap and you look for the bacterium. There you go. New Zealand, hello. Emma, another New Zealander. Besides, boy, this eCam is a pain. Besides Andrew. Oh, you're welcome, Emma. I'm glad they reached New Zealand. What a gorgeous sky behind you. Look at that. Oceans, mountains, clouds, blue sky. Thanks, Emma. Enjoy. Frank, you want a ticket? Okay, I am going to put you on the list right now so that we don't sell out. Let's see. Twiv 1000. Frank wants a ticket. You are 112. And one ticket, and I will email you the details for that. 112 tickets sold. That's very exciting. I think that's cool. What airport is the best? I think it's much easier to get into Newark and take. So in Newark Airport, you would fly in, you would take the monorail at the airport, which brings you to the New Jersey Transit station. And then you get on that, and it's a couple of stops. You're in Penn Station, New York, which is like 6 blocks from the theater. It's 2 blocks from the incubator and 6 blocks from the theater. If you fly into JFK, first of all, I don't like JFK. And secondly, you can get to New York on a subway. It's just a little more involved and a lot longer. So I would go into Newark. Yeah. Will you ever give a tech tour of your video setup and production procedure? Well, you know, I would do that. Yeah. Yeah, I would do that. Any of you watch Marcus Brownlee, his channel? He does tech reviews. So he has a studio in, what's that town next to Newark? Oh, come on. Kearny, Kearny, New Jersey. Oh, what a beautiful studio. He has 3 people helping him. He shoots beautiful videos. So I could do it myself, but I'd rather have someone help with the shooting. It's a lot easier when you have someone setting up lights and camera angles and that sort of thing. It takes a fraction of the time. But yeah, I'd love to do that because we have a thing that we do. So yeah, for sure, Paul. I mean, I've learned from everyone else, but it's an interesting mishmash. Do I think the recent Marburg episode poses a risk? I don't. I think the risk is quite low because these viruses, Marburg and Ebola viruses, while they can be quite lethal, right? High case fatality ratio. They don't transmit well from person to person. You know, they transmit typically in families, close contact, or if a family is preparing a body for burial, of course, they're going to contact it, or in hospitals where they don't do proper infection control. But if you do the right procedures, it doesn't transmit at all. And if you take care of patients in a certain way, they have a better chance of living or of surviving. So I don't think it's a threat, no. So, you know, someone could say, well, how do you know it's not going to mutate to become more transmissible? Well, I don't know. But it hasn't so far. And I'm encouraged by that. You know, the ones that are a problem are the ones we don't know about, right? Someone said last week, when we watch something like H5N1, then it doesn't happen. Do you think the Des Pommiers Quartet can perform the French horn quartet that was written for your award ceremony? I suppose so. Don't jazz players read music? I have the sheet music. It's just that it's written for 4 French horns, right? So I guess there are 4 parts. I don't know anything about music, but on the sheet, I think there are 4 parts, right? 4 lines in each staff or whatever it is. So I don't know. It is a quartet, so I don't know if the drum could play one of the French horns. I'll ask Randy. That's so funny. How am I going to remember? Oh, I'll write it in my... So the French horn, so it's CD 155 is the name of the piece that they wrote. Does everyone know this? Does everyone know that when I got the award last year in Zurich, which is the Richard Ernst Award, they made they wrote a piece of music in my honor, and they called it CD155, which is the name of the poliovirus receptor, which was discovered in my lab or identified. And the first 5 notes of the piece are CD155. Isn't that cool? CD155 for Des Palmiers Quartet. Okay, we'll find out, to listening one. I was reading Breathless. Good book. It has one theory of evolution, that viruses descended from cells that lost function. So I think viruses, and Kunin would agree with this, they have multiple origins. One origin is that there was an RNA world, pre-cellular RNA world, with just molecules in the environment self-replicating, and they were RNA-like. And they eventually became cells, and then the RNAs went into cells for a better replicating environment. But then as cells got more complicated, certainly pieces of the cells could leave. Nucleic acids could capture a capsid and leave. And so the minimal replicon could be a DNA encoding a protein that orchestrates the host to reproduce it in a capsid protein, like SV40, right? But what a virus would need to gain a cell, I'm not sure that, well, so the RNA world theory, yes, it posits that a virus became a cell, but it was a long, tortuous road, okay? So listen. We have RNAs in solution in the early pre-cellular world, Earth. They're replicating. There's no proteins, okay? So eventually to get cells, you have to have proteins. So somewhere along the line, you have to have the protein synthesizing machinery arise, which is plausible because the ribosome, even though it's made of RNA and protein, the catalytic part of the ribosome, the part of the ribosome that actually makes proteins, is the RNA. It catalyzes the bond. Protein has a scaffolding role. So it's possible that ribosomes arose early before proteins, and then they had some other function, and then they were co-opted to make proteins. And then once you have proteins, then you could start thinking about making cells for which you need membranes. So you have early cells. Maybe they were RNA-based, and you needed probably an enzyme to reproduce the RNA. So proteins arise, you get polymerases. Actually, the first proteins that we think arose were RNA binding proteins. And those could then change into polymerases. And then you need polymerases, you need protein synthesizing machinery, presumably you need membranes, so you need lipid biosynthesis enzymes. So it's pretty complicated. But you're limited in size. You only get to a certain size with RNA genomes. If you want to get more complicated, you need to have DNA. So how does that arise? Well, the RNA polymerase, one amino acid change could probably make it into a DNA polymerase, or a few. And now you have DNA, and it can get bigger and more complicated. So that's what I think is going on. Dr. Soup, yep, isn't that good? Could you describe COVID origin evidence that is in the genome of the virus as opposed to looking at geographic evidence of where first occurrences occurred? Okay, well, that's fine, but the geographic evidence is really strong. But the geographic evidence also includes genome evidence. There were 2 SARS-CoV-2 genomes circulating early in the marketplace, and that means 2 different spillovers. However, let's go to the phylogenetic tree. If you take all of the coronaviruses that we knew of at the time of identification of SARS-CoV-2, you build a phylogenetic tree, which means it has a root and branches and a trunk. It tells you how virus genomes are related to each other. And SARS-CoV-2 was not very close to SARS-1, but there were a lot of bad viruses that were closer. including RATG13, a bat isolate, or actually a bat genome, a coronavirus genome sequence from bats. Very close, but not close enough to be the ancestor. So for SARS-1, you see a similar situation. You could actually find the ancestor of SARS-1 in bats with almost identical genome sequences, the virus that was in people. And then subsequent to the pandemic starting, people went out and did more looking for viruses in bats. And they found even closer viruses with closer genomes to SARS-CoV-2, indicating that these viruses are out there and evolving ways to bind to human ACE2, the human receptor. We even found them in pangolins. Pangolins weren't the source. They got infected out there. I mean, they weren't even from China. They were from Malaysia, the ones with the closest virus to SARS-CoV-2. So obviously there's a source of a very close virus to SARS-CoV-2 out there that is infecting pangolins and eventually humans. We just need to find it. There's no evidence that anyone had that in the laboratory. And as I said earlier, that would be what I want for you to say it came from a lab, to show that somebody was working with it. Maybe not even the virus. Maybe there were some bat tissues that had it in it. But they didn't have it because they reported all the sequences they had from bat tissues, some hundred, some odd sequences, and that wasn't there. So I know it's probably a little complicated, but ask more questions if you want to know more. Okay, now we're getting through the bobbleheads. Vincent will have to wear a lab coat on lives from now on. I threw them all out ages ago. We are going on spring break. Where do you want to go? You know, what do you think of this? I sell excursions where I go and teach people virology. We go on a boat, or if you don't like a boat, we could go on a train, or we could go to a resort, and we could sit around the pool and learn virology, or sit around anything you want. Think people would pay to go on a trip like that with me? You know, I know people who do that, like Leo Laporte, who does the This Week in Tech podcast that inspired me. You know, he does an Alaskan cruise where he goes and people go because he's on the cruise and they could talk to him and he gives talks. I could do that. I could do a cruise and teach a course, and we could chat afterwards. Would anybody want to do that? Nah, nobody wants to do that. Okay, never mind. According to Casadeval, antibody levels to spike predicts clinical prognosis. Now, actually, what he said was bulk antibody levels, not even spike antibody levels. Bulk antibody levels predict the effectiveness of convalescent plasma in taking care of COVID if given early enough after infection. So bulk. I'm kind of surprised by that, frankly, because you know, the SARS-CoV-2 component of that bulk antibody is pretty small. So I am surprised, yeah. Racaniello 2023. Oh, boy, we're getting good stuff. This trip on virology. Oh, this could be fun. I might just do it. Get me out of the incubator for a week, right? We have to do it when the incubator is self-running. Right? Thomas says, I could put your bobblehead next to my Tony Fauci head on the bookshelf. So that would be an honor, right? I bet they sold a lot of Tony Fauci bobbleheads because, you know, millions of people know him. Only, I don't know how many people know me, 100,000, 200,000? Hello, Carol, our nephrologist. I'll never forget that, from California. What do you think animal agriculture, especially factory farming, increases the risk of a pandemic. Well, I do, but there are a lot of birds out there already, and they are infected. They can be infected with H5N1. So I think the contribution of animal agriculture, factory farming, is that it brings humans and the birds in close contact, right? Because all the people who have to run the farms are quite next to the bird. In fact, many of those cases of human H5N1 that I talked about, they occur in people who have chickens. In many countries, they sleep with their chickens, right? They only have one room, and the chickens are in there with them, and they inhale their virus and makes them sick. So it's for sure that massive farming increases the risk of getting infected. A pandemic, honestly, I don't know, but what are we going to do? We're not going to stop that practice. We're not going to stop factory farming of chickens and ducks and whatever else, turkeys. We have massive turkey farms here in the U.S. Not going to ever stop, just like we're not going to stop pig farming, and that's a big contributor to other influenza viruses. But I do think, yes, that the more a virus replicates, the more likely it's going to change in a way that could make it transmissible in people. But I don't see that we're going to do anything to limit it. If they immunize the birds, that might help, but I don't think farmers like to do that, right? They're just now talking about immunizing for H5N1, but farmers, if you have so many chickens, it's a lot of work. It costs money and a lot of time and so forth. Since tooth decay is through bacteria, why can't we have a vaccine for that? That is a Michael Schmidt question. I'm going to get Michael Schmidt on maybe after the 22nd, right? The 22nd is going to be Q&A with A and V. So then the 29th, I will see if Schmidt can come on, because you should ask Schmidt that question. He's the tooth decay person. So Michael Schmidt for office hours, OH. I don't know if many of you know Michael Schmidt. I don't know how many of you listen to Twim. Michael's a character. He likes to talk. He gets kind of reined in on Twim, not by me, by others, but here he could talk as much as he wants. So Farhad, I will ask him that. Why does Zika untranslated region bind to a human protein? Well, it probably binds to multiple human proteins, and it is essential for the ability of the RNA to be replicated, to make more RNA. This is common with viral RNAs, that they interact with both viral and cellular proteins. You ask, how does it help with neurotrophism? Well, I don't think anybody knows that. It's just helping with replication. I don't think it's specific for its ability to reproduce, say, in neural cells, which, by the way, the virus has always been able to do since 1947, since it was first isolated. It's not a new property. But these interactions are essential for replication. Hello, Doreen. Thank you for your contribution to science communication. Read today about a zombie virus found in the Siberian permafrost. Is this worrisome from infection potential or just material for another Jurassic Park movie? Well, so the permafrost no doubt has a lot of frozen viruses in it. They, as hosts die, their viruses, as well as them, were frozen. Whether they constitute a threat, who knows? I mean, it could be yes or no. To worry about it, I think, is not worthwhile because, look, they're melting, and as they melt, these viruses are going to flow out. What's going to be there? I mean, viruses of a variety of animals. And maybe viruses that we haven't encountered here, although I tend to doubt it, but if they're frozen, how many thousands of years ago? I know of an old ice core that's 35,000 years old. That's where they got some of these giant viruses from. But I don't know if they're older. How much older and how much of a threat would that be? So I think no one knows. It's a possibility that there are some potentially dangerous viruses in there, but I'm not worrying about it because I think there are other issues we have to worry about right now, and that's not a pressing one. So, yeah, people make a big deal of it. I mean, so far all the viruses they've got out infect amoeba, right? And so in the paper where Jean-Michel Claverie, a French researcher, described some of these viruses from permafrost, he said the real question is, no, that's not a French accent, sorry. The real question is, do they infect mammalian cells? But he said, I don't want to do that experiment because then I'll be blamed for introducing the next pandemic virus into the world, which is true. But do they infect mammalian cells? Well, someone should look at it. Maybe they don't. Maybe they're all amoeba viruses. I don't know. So I just don't know, Doreen. I think we really need to look at more. We have a handful of them that infect amoeba, and so I think we need more data. Oh, this is interesting. An Amy bobblehead in cashmere. That would be cool. Purple cashmere, right? What do you think of that, Amy? Do you like that? Amy's not on texting tonight. Sometimes she texts me, but she's not on tonight. So, yeah, far above Cayuga's waters. Yeah, I went to Cornell, and that was the name of an episode that we did. Virology above Cayuga's waters. That's right. Hello, Tom. Welcome. Sitting in the back, being quiet. If you get enough bobbleheads going at the same time, would it be a scientific consensus? It could be. All right, so I'm going to buy bulk bobbleheads, and it's up to you folks to buy them. We'll see if I can sell out 50. I'll buy 50, and then we'll see if they sell out. Yeah, here we go. Scientists did revive, but it only affects amoeba. Right, that's what I just said, except nobody's tested other cells. So it infects amoeba, but first of all, they shouldn't say not people because no one's tried to infect people with this virus. We don't know if it infects people. All right, Nicole is working on these lectures. I finished the lecture on viral entry where you describe how viruses move on microtubules. Well, diffusion wouldn't work. That made me wonder, do bacteriophages diffuse because bacteria are smaller? You mean within the bacteria? No, I don't think they diffuse. I think they have their own kinds of motor proteins and filaments as well. So I don't think they diffuse at all. No. With the rapid succession of variants, do you foresee more variants with minor fitness improvements? I do. It's easy. That's an easy prediction to make, right? It hasn't stopped. So, yeah, it's just going to continue. Do you want to guess the chance of a big variant change? Why would you have a big variant change? This is, the virus is percolating along, doing quite well. It's infecting a lot of hosts, which is what viruses do. It's changing to become mainly immune evasive so that it can get through and infect over and over again. So I don't know why a big change. I guess it could be an accident, right? That made it have increased virulence, but that doesn't seem to be something that's selected for. And I always like to think about selective forces. And I know on this stream a while ago, people said, Vincent, not every phenotype is selected. Many are accidental. I got it. I do get it. But I think it's of low possibility. With 1918, there weren't increasingly virulent variants emerging. It was just one that moved through the population. So I don't think this is in the cards. Unlikely. I would say unlikely, yeah. Let's put it that way. Yeah, I know Jeffrey Sachs. I mean, what the hell does he know about viruses? Come on, Jeffrey Sachs. I'm not gonna insult him. You are moving out of your lane, but you should stick to economics. And I don't do economics. I don't even pay attention to my own economics. I pay attention to viruses to the detriment of a lot of things, but hey. David Quammen. On Clorox, this is great. I see. I have a Lysol spray in the bathroom that has the same thing. On Clorox wipes, it says it kills 99.9% of viruses. What are the 0.01% viruses that it doesn't deactivate? Can't kill something that's not alive. You're absolutely right, Carlos. So I figure they throw 1 million PFU at it, and it inactivates 99.99%, which may be the limit of detection of their assay, right? Because you can't do a PCR assay for this. You need to do an infectivity assay. And so you have a LOD, a limit of detection, and that's probably that 0.01%, I would guess. Or there may be some that are not inactivated just because it wasn't long enough or it's not concentrated enough. But, in a virus population, there are always extremes of phenotype, so that could be it as well. But I always find that interesting because, they test influenza virus, a rhinovirus, maybe something else. And, yeah, it's not 99% of all viruses, but 99.9% of what they put in. Yeah. My virology professor at SDSU, San Diego State University, believes that. Well, it's not a belief system. It's about scientific facts. And the facts just aren't there to support a lab leak. They are not there. You can make stuff up. You can make conclusions that aren't based on facts. Nobody is going to stop you because this is a free country and we have a press that can say whatever it wants, and we have a House run by people who cherry pick the witnesses they want. What can you do? It's very sad. And watching this as a scientist is discouraging because, and if Amy were here, she would say, "Oh, Vincent, come on." But when I was trained as a scientist, I thought it was a pure profession. It's not pure at all. You have people doing all kinds of things that aren't right, but then now we have non-scientists messing with it. Oh my gosh. So now we're coming to the questions about the lab leak. Isn't there a preponderance of data that supports zoonotic spillover? Of course. I pretty much went over it at the top of the show. Lineages tracing back. Very closely related viruses in nature, which weren't in anyone's laboratory. The precedent of other viruses. Nicola says, as someone who has spent more time than I'm willing to admit on the history of nukes, I'm super skeptical about anything that DOE says without releasing the actual info. So isn't that a good practice? You say, oh, we think this happened, but we're not going to give you the evidence. Oh, what kind of crap is that? The only conclusion I make is that you don't have any evidence if you don't want to show me. Because if I'm a scientist and I say, this is the way it works, I will show you the evidence. So they're not working from a scientific basis. Laurie Garrett tweeted today about a significant percent of Australia's bat population dying from heat and subsequent potential disaster that may ensue without them. Let's look. Australia, bat die-off. die off. Let's see what we got. In the last 2 decades, tens of thousands of Australian flying foxes have died during extreme heat events. They are arguably among the most dramatic mass mortality events witnessed in nature, but they can be indicators of heat stress and more cryptic fauna where impacts are more difficult. So this has been going on for a while. They've knocked out a lot of a third of bat species. So this is a real thing, yep, but it's not anything new. I mean, I'm looking at stories here from 2018, 2019, 2015, 2014. So, yeah, this is caused by heat. And it's not good because bats are highly beneficial, right, to the ecology. The insectivorous bats eat a lot of insects. Actually, I asked my guest at the bat meeting this last summer, what do bats do? Pest control, pollination, dispersing seeds. Bats eat enough pests to save over $1 billion each year in crop damage and pesticide costs alone in the U.S. So, yeah, we don't want to lose them for sure. Even though at the beginning of the pandemic, if you remember, people wanted to get rid of all bats because that's where the virus came from. I guess now they don't want to get rid of them anymore, right? Because they're thinking it didn't come from them. They're thinking that, not me, right? Question for Dr. Griffin. I'm a geezer. Pharmacy refused PVC vaccine script because I'm much older than 45. Why should active old people be refused a vaccine protection? You shouldn't be. Sorry. You know, there are different practices all over the place. You're talking about the pneumococcal vaccine, right? I don't know why they would refuse that. I'm sorry. Go to another pharmacy. They shouldn't refuse that to you. But if you want me to ask Dr. Griffin, I will. PVC vaccine refusal by pharmacy. If we have any pharmacists here, could you weigh in? That would be nice. Could a laboratory create a SARS-type virus? So what do you mean create it? From what? From coffee beans? From what, Dennis? Tell me what you want to start with. If you mean from scratch, if you say, let's make 30,000 bases of RNA that codes for a virus that has these properties, no. No. Can't do it. And remember, if you want to make SARS-CoV-2, you couldn't do it from scratch. You'd have to have something close to it, and we don't have anything close to it. So the answer is no. You wonder if they're ever going to get nasal vaccines approved. Well, there is one already approved, right? Flu mist. You're talking about SARS-CoV-2, right? Well, if it keeps infecting at the current rate, yeah, people will continue to work on it. But remember, any new product needs to have a market that will sustain it. Especially development costs have to be recaptured. So and that's what someone wrote in to TWIV, and they said, people are asking why new monoclonals aren't coming out. Well, by the time you develop a new one, and if it's already, if resistance to it has already arisen, there's no monetary incentive to do that. So that's why we're not seeing new ones any longer. Forces in the government might want to smear China right now. It seems the more convenient motive than actually having new evidence. If they had a progenitor, wouldn't they make it public? I would think so, because scientists would want to know what's going on. They don't have a progenitor, John. They just have hearsay. They say, oh, there's the Wuhan CDC next to it, so it could have come from there. Yeah, okay. So show us some evidence. Oh, she said, so bad cat meant HPV vaccine. Oh, you're older than 45. They won't give it to you because it's of no good. It's of no use for you. You're probably already HPV positive. You're already HPV positive. You can't. Yeah, it can't be given to people over 45. That makes sense now. Oh, so Amy watched the hearing. It was despicable. In what sense? Can you give us a little detail? Intelligence services don't usually release all their data. Well, that's a poor excuse in this case, Claire. Sorry. If you have scientific evidence, you better release it, because you're never going to convince anyone. And I would say they are under the obligation to release it, because just telling me it came from a lab, I don't believe them, and you shouldn't either. That's just nonsense. Thank you, Carmen, for your contribution to science communication. Wouldn't a lab leak create a different outbreak pattern, random cases throughout the city? I think they would be focused at the WIV, right? Just like they're focused at the Huanan seafood market. If you look at the earliest cases, they're at the Huanan seafood market, not at the WIV. But I think if it spilled over from say a carcass at WIV into people, that would be the epicenter, and it's not. Yeah, that's the way epicenters work epidemiologically, right? And Peter says if they don't release it, it isn't data. That's absolutely right. And you can say all you want, but we don't believe you. Topol was on UCSF with Bob Wachter. He thought the COVID nasal vaccine research was going well. Well, I'm glad he thinks they're going well. These are two people I'm not going to continue that sentence. I think just take what they say with a grain of salt. Can spike proteins be modified to attach to specific membrane proteins? Yeah, they could be. Absolutely. Now, that doesn't help you, but yeah, we can retarget membrane spikes to hit different membranes for sure. Absolutely. We do that often for modifying vectors that are used in various cases. Yep. Did the wet market vendors want to cover up the illegal wild mammals? No, the vendors didn't. The Chinese government did. See, they did not want the world to know that they screwed the world over by keeping these markets open. That's my take, which isn't worth anything. I agree. I'm just telling you because this is my live stream and you want to hear what I have to say, right? I don't think they care if you make rumors up about the lab, because labs do research. That's what they do. But the markets were supposed to be closed, and they weren't. And as a consequence, a virus got out there, and it screwed the world over. I'd rather use a stronger word, but I'm going to use screwed. And it's their fault because of the market. They know it. So on January 1, they went in and wiped it out so that very little evidence is left to make a definitive isolation, say, of the virus. But we have enough in terms of the epidemiology. We do have some environmental sampling in the places where mammals were kept. So it's as good as we're going to get, as Michael Warby says. But it wasn't the vendors. It was the government who did that. Has H5N1 gone human to human, not extended chains of infection? No. There have been some human-to-human in a family where chickens infect some, but I think you have to be careful about those because you actually don't know it's human-to-human as opposed to two independent events. So, no, not yet. I'm wondering if when they say waning, is it actually waning or just immune evasiveness? Well, I don't know what you mean, Brittany, because there's not a lot of context here. But, well, it's a combination, right? You could do waning if you did binding. If you say, let's look at all the antibodies that bind to spike, not neutralizing. And you can, that way you get around immune evasiveness. Actually, maybe not. No. I don't know who's saying this, but waning is a decline in the antibody level, and immune evasiveness is a decline of the neutralization activity. Do your students also take a class on the other microbes? Yeah, there is a microbiology class at Barnard, but there's not much down at Morningside, no. And I don't know if my students take it. I actually don't know. Oh my gosh. Redfield said today that gain-of-function research unleashed a new virus on the world without any means of stopping it, resulting in the death of millions of people. Oh, this is just... He's gone off the deep end. Oh my gosh. Of course, he said what they wanted him to say. They said to him, you should say this. This is just an embarrassment. This was the head of the CDC. There's absolutely zero evidence that anyone did this. I mean, we've told you over and over again that, I mean, now he's confusing the two theories, right? The lab leak versus, lab made or lab origin. No, that's not it. It's a lab leak where it got out versus man-made or human-made. So he's not even on the right program. Nobody made this virus. We got rid of that theory a long time ago. The only thing that's on the table, in my opinion, is whether there was a carcass in a lab somewhere that somehow infected someone. And I would say there's zero evidence for that. But this making the virus is absurd. Totally absurd. And that's been gone through multiple times all over the place. I'm just stunned that he would say this. Oh, my gosh. What's your thought on the bivalent in seniors? I guess you mean waning. Yeah, I'm not surprised. The seniors don't mount a sustained antibody response. They're not good at making memory, which is what sustains your responses. So I just think it's a problem to depend on the vaccine to prevent you from getting infected because it's only going to last a couple of months at best. The antibody levels are going to contract. I think you need some other plan like Paxlovid or Remdesivir or Molnupiravir is way down there. I don't think you should be getting a frequent booster to keep your antibody levels up if just for 2 months because I'm not clear even when this virus is going to be seasonal, right? It's infecting all year round still. So you can't even time it. I mean, if it were a winter virus, I would say, okay, let's get you vaccinated in the winter time like we do for flu, but it's not even that. It continues afterwards. So this is a good point. People wedded to the lab origin move goalposts until they finally say that Zhongli, Xi, and CCP covered up dangerous research, so we'll never know. They pose an unfalsifiable theory. Yes, of course. That's their data. They were doing this, and it's covered up, so we won't know. In the meantime, we get a lot of data on natural origin, or I say zoonotic origin, and they ignore it. Leventhal's paradox. I don't know about that. Cy Leventhal? Let me look it up. Yeah, thought experiment. Yeah, it was Cy Leventhal. Whoa, look at that. I knew it was Cy Leventhal, at least. In the theory of protein folding, he said, because of the very large numbers of degrees of freedom in an unfolded polypeptide chain, the molecule has an astronomical number of possible conformations. 10 to the 300th. Okay, therefore, if a protein were to retain its correctly folded configuration by sequentially sampling all the possible conformations, it would require time longer than the age of the universe. So that's the paradox. He was aware that proteins do fold spontaneously and on short timescales. He suggested the paradox can be resolved if protein folding is sped up and guided by the rapid formation of local interactions, which then determine further folding of the peptide. Frankly, I don't know why it's an issue. We know that proteins fold, so there is no paradox. It's sorted in some way, right? I wonder what Jens Kuhn has to say about the lab leak. He won't say it probably because he's a government contractor. But who knows? He might. He's not listening to this. He listens to TWIV. He might be vocal about it, right? He tends to really speak his mind. I like that about him. Okie dokie. Can you share your thoughts on Draco? Well, Draco was a broad spectrum antiviral. Draco TWIV. Let's see. We did this 146. It was called Draco's Potion, right? A little reference to Harry Potter, right? Let's see. Broad spectrum antiviral plus. We developed a new broad spectrum double-stranded RNA activated caspase oligomerizer that induces apoptosis in cells containing viral double-stranded RNA. Yeah, so they put together a modular protein. The problem with that is that it's a protein, and trying to get proteins into cells is not easy. So that never went anywhere. Probably for that reason. So I say that if we did have a broad spectrum antiviral, it could be a problem because I think many of the viruses we have in us are beneficial. We just don't know it. We don't have good evidence for it, but they're there. Many of them are there, and they don't seem to hurt us. So maybe they're helping us. And a broad spectrum would get rid of them. And that's actually the experiment that would tell us if these viruses are beneficial or not. Of course, we would know this long before such a drug were licensed, right? We would know that in a trial it had safety issues, right? So it would never be used at a mass scale. But it's like wiping out your microbiome, right? You get sick. When you take antibiotics, you get dysbiosis, you get unbalanced bacterial communities growing back. It's not good to do. We do it for a variety of reasons. If you need surgery, they blast you with antibiotics, and sometimes the outcomes are very poor. Can you explain a little bit about satellites, helpers, and hepatitis B? So a satellite virus is a virus that can't do it on its own. It encodes some part of the virus, but it requires a helper virus to infect the same cell. And so, for example, in some cases, let's take the hepatitis delta virus. It requires to be in the same cell as hepatitis B virus. And the hepatitis B virus provides the capsid for hepatitis delta. Delta has its own genome, which encodes for one protein, the delta antigen, which coats the viral RNA. And then the hepatitis B provides an envelope. So that's just one example. Delta is a satellite because it requires co-infection with Hep B. There are lots of examples of that. Adenovirus-associated viruses are helper dependent, let's say. They get a different name, but it's a similar principle. They can only reproduce in cells co-infected with adenovirus or herpes viruses, which provide a number of functions that AAV requires. And there are a whole host of plant viruses and insect viruses that are satellites. There are the giant viruses have little viruses in the same cell that depend on them for reproduction. So there's a lecture in the course devoted to unusual infectious agents way at the end, where we go into them in more detail. But that's the 35,000-foot overview. I had COVID a month ago, severe flu-like symptoms, no fever. Was disappointed because fever helps fight infections, or does it for viruses? I had a plan and I got Paxlovid. Very good. Fever is a consequence of cytokines that are produced during an infection. And, we like to think that high temperatures impair microbes, but I think in the case of viruses, that's not really correct. I don't think it's been proven. I think the fever is a result of the cytokines. Why it happens, it may have more to do with bacterial infections. Why do we have fever? Virus reproduction, the Medline Plus will say that your body is trying to kill the virus or bacteria. But, you can grow viruses in the laboratory at relatively high temperatures, and they do very well. So what is the temperature? So what is a bad fever? 105 Fahrenheit, right? 105 F in Celsius. So that's 40 degrees Celsius, 40 and a half degrees Celsius. We used to grow viruses at 39 and a half in the lab, and they're fine. So I think this is wishful thinking, frankly. And if you don't have fever, you didn't have the right mix of cytokines. I bought the textbooks. The material is more complex than your lectures. I'm lost. It's getting worse. I'm sorry. I'm giving it the old college try. Yeah, my lectures are a distillation of the textbook, right? And it's simplified because you can't put it all in a course. It would be just too much. But I'm sorry it's hard. But step back, maybe listen to the lecture and go back and take it in chunks. And if you have a question, email me. I can start you on the right road. Vincent@microbe.tv. If people are weak in genetics, Thomas Minella on YouTube offers an excellent full course. Let me see. Thomas Minella, I'll write that down. Check it out. Thank you for that. Post and Ghost. Is that the way it's called? I like that. I just don't. I'm not going to get in a fight with people. It's just not worth it. They're not going to ever agree. I'm not going to teach them anything. Nope, not happening. Not worth it. I have. What was I going to say? I'm looking at comments and I lose my thought. I forgot. I have engaged in the past. It's not worth it. I have other things to do. I have more things to do that I haven't been able to do, and I'm certainly not going to take some of that time and spend it on Twitter. Time magazine just put online people who get Omicron are less likely to get long COVID. Put out, yeah. Well, I'd have to look at the study. I'm a little skeptical because these are very difficult conclusions to make. Omicron is not mild. I don't know why they're saying that. Plenty of people, the people who are dying now, and people saying, "Oh, look, so many people are dying of COVID." They're dying of Omicron. Come on, right? It's not mild, so I don't know why that would be. The propensity to attack those we don't agree with is sad. It's very sad. I don't know why you have to attack. Just make your point and say, what do you think of that? You know, I heard that data is a dimensionless word. What do you think? And I'll say, well, here's where my source is, that there's a singular and a plural. Oh, thank you for pointing that out. I didn't know that. That would be a civil discourse that leads to somebody learning, right? But we can't do that anymore. Part of it, I think, is the anonymity of posting, where nobody sees you. If you went up to someone and said, "You're a jerk. Data is a non-dimensional word," they wouldn't. Boy, it would take some character to do that. Right? There's some people out there who would do it, but it's a problem with writing a comment. Let's see. You like the piano behind me. There's no piano behind me. No, there's a guitar. There's this. That's not a piano. Those are speakers actually that are not working. They're just stacked there. Where's my guitar? It's over there. You can't see it. It's dark. There's no piano here. It's funny. When debating a serious topic, I believe it's best to be not dismissive or obtuse. I agree. I try not to be, right? So the wool of polio was spent 6 well plates. Yeah, exactly right. What kind of plaque has it? What cells did you use? Those were HeLa cells. They make nice plaques. Yeah. I don't know if it's still there. I bet it is. Because whoever moved into the office probably didn't bother to throw it out. I'm beginning to get adverts for life science laboratory equipment on my YouTube feed. I blame Vincent. This is my fault. Yeah, you're watching science. So that's what they're doing. Should we be concerned for your safety with these attacks on your statements of facts? I don't think so. I'm not concerned. I'm not worried at all. I think these people are cowards and they attack you on paper or online, and that's about it. So I appreciate the concern, though. Thank you. Appreciate it. Twiv is the first place I go for COVID info. Thank you for always sharing. You're welcome. And all my co-hosts, of course, are great as well. They come with me every week. What happened? I think it's still there. I left it behind when I moved, Rima. And I think it's still there because probably whoever moved in didn't want to bother clearing it out. Is that like the triangle of sadness? I don't know what you mean. The wall of polio, the triangle of sadness? I don't think so. It's very different. Have you thoughts on this case report? Clearance of long-standing immune deficiency associated vaccine-derived polio infection following remdesivir therapy. Oh, that's very interesting. Let's see. Remdesivir polio. Let's see if we bring it up. Oh, look, here it is. Frontiers. Clearance, yes, as you just said. That's quite interesting. Strong justification for further clinical studies of remdesivir. So the problem is that the oral polio vaccine, the infectious one, if you give it to immunocompromised people, they reproduce the virus for many, many years. And we don't know where they are because they don't get paralysis and we don't screen for them. But if we do identify them, there's no way to stop their infection. So this suggests that remdesivir may be a way to do it. That's very interesting, Spar. Thank you for bringing it up. Very cool. I have to show that to Amy. Do you hear, Amy, about that? No, not listening. Okay. My friend is 80. She got COVID. Did not take Paxlovid. Well, she's lucky. Okay? Lucky. I'm glad she's okay. Because when I'm 70 and I got COVID and Daniel said, you're taking Paxlovid. I said, let's see what happens. He said, no, we're not going to see. Why can't you and fellow scientists write a letter so the times are similar, articulating why you're not convinced? I think the times is not into the lab leak. I think you're going to be seeing something coming out soon which refutes that. So I don't think we need to do that. I mean, Amy and I wrote an opinion piece, which was totally ignored, a year ago, or a year and a half ago, about why it's not the variants, it's the people's behavior that's spurring the spread of variants. But nobody wanted to listen. Everybody has their own agenda. Thank you, JJ, for your thanks for public education. We love to do it. I would love to come down to New York for your shindig. Get a house sitter. I would sit your house, but I can't because I have to be at the shindig. Sorry. Frank says, thank you for providing this information in an accessible format. You're welcome. I'm glad we could help out. Slavitt and Garrett were on Al Franken's podcast. They never get it quite right. Well, Slavitt never gets it right at all, so at least with the science goes. He doesn't know what he's talking about. No, they don't want to talk to me. So Slavitt and Garrett are more public figures than I'm just a little podcasting professor from New York City. It's okay. What is the word? I'm patient. I am patient. Okay, so back to Bad Cat. So they say, okay, you may tell us that you only had one partner, but we're not going to make an exception for you because the blanket rule is 45 and older. But I will ask him. I don't know if I can do it tonight because I have too many questions already, and it's 10 o'clock, and I don't want to go past 11. But are there any student tickets? I have to look. Vincent at microbe.tv. What are the consequences of an ambisense viral genome? The consequences are very simple. That the strand of RNA has both plus and minus polarity. So part of it could be translated directly, and part of it has to be copied to a plus strand in order to be translated. It's just another way of organizing genetic information. My understanding with Paxlovid rebound with first cases they tested PCR positive, got meds, PCR negative, but then PCR positive. And yeah, so there's something wrong with the PCR tests. Yeah, the symptoms are from the inflammatory phase. And I think in a certain fraction of people, the viral RNA levels continue at a certain level. They don't go down as much as in others. And so maybe one negative was wrong. I mean, there's no Paxlovid rebound. Do I drink tap water? It's interesting. In the incubator, the water's cloudy. It has some precipitate that falls out. I don't know what it is. It doesn't taste good. So I buy boxed water at the incubator. And here I have a reverse osmosis system for the water. But I would drink the tap water here. I've drunk tap water in many places. I just, sometimes the taste is strange, and you can fix that by a Brita or some other means. The incubator, I got a Brita, but it didn't take out the precipitate, so I haven't done it. Okay. It's hard to ask a question in 200 characters. I was wondering if a modern virus could regain the capacity to self-replicate if they ever lost it. Yeah, like a flightless bird learning to fly. Well, we don't see that happening, right? So there has to be pressure for something, selection for something to happen, right? So if the genome has a polymerase, you lose the polymerase, it's not going to replicate because it needs some other features to self-replicate, and I don't know how you would get those back. So I think that's highly unlikely that would ever happen, right? That's right. Nearest known ancestor is Banal, 2052, found in Laos with 96.8% similarity, 960 nucleotides difference. Still away. It's not the ancestor of SARS-CoV-2, but it's very close, and it's in Laos. It's in Laos, okay? It's not in China. These viruses are all over the place. We haven't done a lot of sampling, and so we could find more. Couldn't they have just gotten the virus when they collecting bat samples? They could have, but they didn't have it. That's the point. They didn't have it. They published their collection. It wasn't in it. Anyway, they don't even have viruses. They have genome sequences. They can't get viruses out of most of these samples. There's not enough material present. They do genome sequence. And so the people will say, oh, but there's an exception. Yeah, there's always an exception. Sure, run your science life that way, that there's always an exception. See how far you get. Oh, my gosh. I don't mean to pick on you, scientific libertarian, but they didn't get the virus when they were collecting bat samples. They get vanishingly small samples. They usually throw them into a solution that inactivates the virus. And if they want to keep some for virus isolation, they never get virus from these samples. All right, I have to start wrapping up because Daniel Griffin's working, waiting for me. And I just got to the point where we were talking about Virology river cruises. Oh, and I didn't get to see it. So we're going to have to skip it over because I want to thank everyone who have donated. Thank you, Jana King, for your contribution to science communication. Oh, my God, there's so many great ideas for a river cruise. There are travel agents who will put you together such a tour. Let's go to the Arctic and Greenland. We can do that, and we can learn virology on the way. I would absolutely do this, folks. If you're into it, I'll look into it. I don't mind. I don't like cruises, that's for sure. But for you guys, I'll take a cruise and we'll learn virology. Absolutely. Totally. Yep. We'll be safe. Don't worry about it. That's great. All these comments, I'll read them tomorrow. Yes, viral file. Where is it? Viral file passengers sickened by viral disease on this trip in virology cruise. That would be funny. Maybe we'd get to the headlines finally. All right, we'll do it. Looks like everybody's into it. Thank you, Gabriel, for your contribution to science communication. I'm sorry I have to rip through. I would actually go a little longer tonight if not for Daniel waiting for me. 361 people tonight, 299 likes. That's great. A lot of good questions. Holy cow, folks. I'm very impressed that you're still here and you're still asking questions. Raka Pedra, thank you for your support. Is it something to worry about? If you're generally healthy and young and you have 3 vaccinations, you're good. If you have other illnesses, if you're over 75, you need to have a plan to get an antiviral if you get tested positive. Okay. These are great comments at the end. I want to read them, but I can't do it now. Okay, folks. That'll do it for another office hours. Thank you all for coming. You were great tonight. Great questions. Great liveliness. You really keep me going here. And let's continue the conversation in 2 weeks. Remember, we're going to have Q&A with A and V in 2 weeks. No office hours next week. Take a break, everybody. I want to thank our mods, Frank, Les, Tom, PDK, Barb Mack, and Vanity Nutrition. Thank you all for coming tonight and moderating. Thanks all of you for coming and bringing your questions. Really appreciate it. If you want to go to Quiv 1000, Vincent@microbe.tv. If you have any questions, Vincent@microbe.tv. If you want stickers, here we go. If you want a Microbe TV sticker, tell me, Vincent@microbe.tv. I'll send it to you for free. I just need your postal address. I love you all, folks. Thanks for coming. Have a great night. Be safe. See you next time. Good night.